Our presentation today will describe general principles of product-specific guidances, or PSGs, discuss the process of how PSGs are developed and revised, describe how PSG revision efforts are prioritized, and share a recent example of significant PSG revisions, and describe the upcoming PSGs for complex generic drug product development webpage. The Federal Food, Drug, and Cosmetic Act requires that an applicant submitting an abbreviated new drug application, or ANDA, to FDA for approval of a new generic drug product show that the generic product is, among other things, bioequivalent to the corresponding reference-listed drug. But how do you know the best current bioequivalence, or BE, approach for a product? As a starting point, the regulations governing bioequivalence are provided at 21 CFR Part 320. FDA also publishes general guidances for industry, such as the bioequivalent studies with pharmacokinetic endpoints for drugs submitted under an ANDA guidance. And FDA also publishes product-specific guidances to provide recommendations for developing generic drugs and generating the evidence needed to support approval. One thing to note is that PSGs are only applicable to ANDAs and not other types of applications. Beginning in 2007 and continuing through to today, FDA has published PSGs to foster generic drug product development and ANDA submission and approval by providing clear and direct recommendations to ANDA applicants. PSGs are intended to make applicants' research and development decisions more efficient and cost-effective by identifying the methodology and evidence recommended to support a specific generic drug's approval. They help applicants submit ANDAs with fewer deficiencies. And PSGs are a key science and research output for FDA. As of March 2020, more than 1,800 PSGs have been published. PSGs, along with general bioequivalence guidance issued by FDA, provide FDA's current thinking and expectations for generic drug development. Consistent with 21 CFR 320.24a, the scientific recommendations reflected in PSGs represent FDA's current thinking on the most accurate, sensitive, and reproducible approach for conducting BE testing. As mentioned before, FDA publishes PSGs to foster generic drug product development. Under the GDUFA II commitment letter, FDA will issue PSGs for 90% of non-complex new chemical entity NDA or new drug applications that are approved on or after October 1st, 2017, at least 2 years prior to the earliest lawful ANDA submission date. I'll give an example of what that time frame looks like in a couple slides. For complex products, as defined under the GDUFA II commitment, that time frame does not apply. Rather, FDA strives to issue PSGs as soon as the scientific recommendations are available. Each PSG will contain the recommendations for that particular product. Under the recommended studies, it will include the study subjects, for example, general population or patients. It will also include the study design, for example, single dose or multiple dose, two-way crossover, partial or full replicate, or parallel. The type of study, for example, fasting or fed. The PSG may also contain additional information, such as analytes to measure and the dissolution method, if appropriate. There could be a waiver section if applicable, for example, a waiver option for additional strengths, or for a BCS, or biopharmaceutical classification system-based waiver option. The PSG may also include special considerations, such as alcohol dose dumping or sampling approach for drugs with long half-life. And in the bottom left-hand corner, the PSG contains the date that the PSG was issued. And if the PSG has been revised, that date or dates as well. New and revised draft PSGs are generally posted quarterly in batches. On occasion, the appropriate methodology for establishing BE for a specific drug product will be the subject of a citizen petition or other correspondence. When the same BE issue is under consideration in different contexts, the agency will take into account the status of related matters in determining how best to address the scientific issues. This may involve, for example, coordinating the consideration of a pending citizen petition with the development and publication of a PSG, either in one of the quarterly batches or by itself. For non-complex new chemical entity NDAs, as mentioned previously, that are approved on or after October 1st, 2017, FDA's goal is to publish the PSG at least 2 years prior to the earliest lawful ANDA submission date. By way of example, a non-complex new chemical entity NDA would receive 5-year new chemical entity exclusivity, which would run from the time of NDA approval. The earliest lawful ANDA submission date would be 4 years later if an ANDA is submitted containing a paragraph 4 certification to a listed patent. Therefore, the GDUFA goal date for posting the PSG for that product would be 2 years after the NDA approval date, which would be 2 years before the earliest lawful ANDA submission date. And as mentioned before, for complex NDAs, that goal does not apply. Rather, FDA strives to issue the PSG as soon as the scientific recommendations are available. As will be discussed in more detail in later slides, FDA provides advance notice of planned new and revised PSGs for complex products. FDA also develops and issues PSGs based on requests from industry, which can be submitted via email to genericdrugs@fda.hhs.gov, and based on public health priorities identified in the CDER prioritization map as revised. PSGs are posted on the PSGs for Generic Drug Development webpage, which you can access from the link located on the bottom of this slide. You can also navigate to the PSG webpage from the CDER Guidances webpage. From this PSG webpage, you can access and save PSGs. You simply need to click on the link for a particular PSG on the left-hand side. The PSG will then open, as you can see on the right-hand side of this slide. You can browse for PSGs by the first letter of the active ingredient. This will show you all PSGs that begin with that letter in alphabetical order. You can also search by the active ingredient, as well as the reference listed drug number or the reference standard number. If you are searching, you will need to enter at least 3 characters in the search box. You can also type the full name of the active ingredient or all numbers of the reference listed drug or reference standard. You can also see the most recently added new and revised guidances below the search box. BE recommendations are revised as appropriate to ensure that the most up-to-date BE information is available to the public. To ensure that you are using the most accurate, sensitive, and reproducible methodology, please monitor for the availability of revised as well as new PSGs on the PSG webpage and in the Federal Register. The PSG webpage also includes instructions on how to submit comments on PSGs, which I will discuss in more detail on the next slide. Published PSGs, both new and revised, are announced in a Federal Register notice and made available to the public on FDA's website, which was shown on the previous 2 slides. The Federal Register notice is posted in the docket number shown on this slide. The Federal Register notice will identify a comment period for the draft recommendations, which is typically 60 days after the notice is posted. There are instructions in the Federal Register notice, as well as on the FDA website, which explains how you can submit comments. Comments can be submitted to the docket for these guidances or mailed to FDA's Division of Dockets Management. The mailing address for FDA's Division of Dockets Management is included in the Federal Register notice as well as on FDA's Product-Specific Guidances webpage. One last item of note with regard to comments. FDA considers comments on draft PSGs in developing final BE recommendations. I will now turn it over to MJ Kim for the remaining portion of the presentation.
Thank you, Dave. For most products, such as oral drug products, the focus of establishing bioequivalence is on the release of drug substance from the drug product into the systemic circulation. Therefore, FDA guidance on BE studies with PK endpoints for drugs submitted under an ANDA is often referred as the key guidance when PSGs of oral drug products are developed. While this guidance is generally applicable to dosage forms intended for oral administration, FDA also recommends that generic drug applicants consult published PSGs when considering the appropriate BE study and/or other studies for a proposed drug product. Some examples are related to sprinkle BE study, in vitro studies to determine the potential for dose dumping in alcohol, and enteric feeding tube studies. For other products, such as locally acting drugs and transdermal or topical products, there are other considerations when PSGs are developed. For example, FDA guidance titled Comparative Analysis and Related Comparative Use Human Factor Studies is for a drug-device combination product. This guidance includes the analysis of proposed user interface for the generic drug-device combination product when compared to the user interface for the reference listed drug. The other example is the FDA guidance on transdermal and topical delivery system. In addition to recommendations on product design and pharmaceutical development and others, it addresses special considerations for areas where quality is closely tied to product performance and potentially safety issues, such as adhesion failure and the impact of applied heat on drug delivery. As previously mentioned by Dave, new and revised PSGs are posted every quarterly in batches. When new drugs are approved or changes are made to the approved new drugs, we would screen and prioritize which PSGs to develop or revise. Once these PSGs are published, we would collect and review comments received from public, apply research findings from regulatory science research projects, or other correspondence and revise PSGs as appropriate to ensure that most up-to-date BE information is available to the public. As we take a deeper dive into how we create a working list of PSGs, there are ongoing efforts in developing new PSGs as new drugs are approved or changes are made to the approved new drugs, such as approval of additional strains. We regularly monitor which NDAs or supplement NDAs are approved, which ANDAs are submitted without published PSGs, whether there are any public or FDA internal requests or needs for PSG development, or if any scientific research findings are available for PSG development or revision. We then prioritize which PSGs to develop or revise based on their NCE exclusivity status, drug shortage, number of pending ANDAs, how many products are available in the market, and others. We also assess if there are any PSGs with major or significant deviations from the FDA BE guidance, and if there are any newly identified safety concerns. This slide shows the distribution of how many new and revised PSGs were published each fiscal year. For example, in FY 2019, there were more revised PSGs compared to the new PSGs being published. This shows how we have tremendous ongoing efforts in maintaining these published PSGs so that they reflect the latest current thinking on recommendations as new information becomes available. Some examples are related to our recent work on fat and fasted BE recommendations, which will be discussed more in detail in the next slide. Other examples are related to new safety findings or approval of additional strengths or indications with the reference listed drugs. As I mentioned earlier, a recent major effort in PSG revision is related to fat and fasted BE study recommendations. As co-administration of food with orally administered drug products can impact bioavailability, FDA recommends that generic drug applicants conduct a fat BE study for orally administered immediate release drug products, in addition to a fasting BE study, except when the reference listed drug labeling states that these drugs should be taken on an empty stomach, or when serious adverse events are anticipated under fat conditions. However, before the PSG updates, ANDA applicants were asked for a fat BE study as part of the ANDA review if it was determined to be necessary to establish bioequivalence. Therefore, as an ongoing effort to bring PSGs up to the current standard, we recently revised the PSGs of solid oral dosage forms to include a fat or fasted BE study recommendations. In addition to PSGs, generic drug applicants may utilize other available resources to facilitate their generic drug development program. If applicants have questions not addressed in the PSG about the design or analysis of such BE studies, or they have alternative proposals to what is recommended in PSGs, they may submit their inquiries via a controlled correspondence. The pre-ANDA program is another valuable information resource for generic drug applicants. Industry can submit pre-ANDA meeting requests for complex drug products if FDA has yet to publish PSGs, or if they have alternative proposals to what is recommended in PSGs. As briefly mentioned by Dave, FDA provides advance notice of planned new and revised PSGs for complex generic drug products. This is done by launching a web page in 2019 describing FDA's plans for issuing new and revised PSGs in the next 12 months. I'll explain what the complex generic drugs are in the following slide. The goal of this advanced notice is to help generic drug applicants with planning their drug development and support the development and approval of safe and effective complex generic drug products, which are often more difficult to develop. For new PSGs for complex generic drugs, the web page provides information on the active ingredients, the route of administration and dosage form, and the reference listed drug application number. For any revision related, a list of PSGs for complex generic drugs under revision is provided along with planned revision categories. I'm going to go over what these different planned revision categories are in the next two slides. Lastly, this web page is updated each quarter once the PSGs for that quarter have been posted. In terms of what the complex generic drug products are, the GDUFA 2 commitment letter defines them as follows. Products with complex active ingredients, formulations, route of delivery, or dosage form. Some examples are peptides and complex mixtures of active pharmaceutical ingredients. Complex drug device combination. Some examples include auto-injectors and metered-dose inhalers. Other products where complexity or uncertainty concerning the approval pathway or possible alternative approach would benefit from early scientific engagement. One good example is abuse-deterrent formulation of opioid drug products. As mentioned earlier, for upcoming revised PSGs for complex generic drug products, the web page provides the planned revision category and briefly describes the nature of anticipated revision. These planned revision categories are major, minor, and editorial. A major revision is when a PSG is being revised to include additional BE studies or evidence recommended to support FDA approval. For example, adding an additional PET BE study or a sprinkle BE study is considered as major. A minor revision is any revision that is not considered major. This includes, but is not limited to, when a PSG is revised to remove certain studies to provide alternative and less burdensome approaches to the currently recommended studies, to add information on newly approved strengths of the RLD, or to make other recommendations that would not generally result in additional recommended BE studies or evidence. For example, adding an in vitro BE option as an alternative approach or clarifying in vivo study design is considered as minor. Lastly, an editorial revision is a non-substantive change which includes correcting grammatical issues or typos and streamlining the format of existing recommendations. Moving on to other critical aspects of PSG and generic drug development, we have the GDUFA Science and Research Program to advance the science of generic drugs and provide essential tools and information to generic drug applicants. This is done by using the GDUFA research findings to develop PSGs as we gain scientific data and knowledge from the regulatory research science. Therefore, GDUFA-funded research plays an important role in supporting FDA's regulatory advice and decision-making across the generic drug program. I would like to share two recent examples of PSGs to highlight these efforts. The first example is levonorgestrel intrauterine system. The revised PSG for this product was issued early this year and recommends a combination of two studies to establish bioequivalence, an in vitro study and a novel in vivo ex vivo study. These revised recommendations were the result of extensive GDUFA research regulatory science initiated partly in response to requests for clarifications submitted by stakeholders as comments to the docket for the original version of the PSG. The second example is a beclomethasone dipropionate metered inhaler. To address challenges with conducting comparative clinical endpoint BE studies for middle-dose inhaler products, FDA conducted research to help identify and develop more predictive, clinically relevant in vitro methodologies for characterizing the aerosolized particles and their deposition and dissolution, as well as new computational modeling and simulation approaches to correlate these results with the delivered dose measured in vivo. As a result of this research in 2019, FDA posted the first PSG for a solution-based middle-dose inhaler. With that, we will move on to two challenge questions. Question 1. PSG recommendations do not address A, what the active ingredients are, B, what the dosage forms and routes are, C, how pediatric study subjects are studied, or D, which analytes should be measured. The correct answer is C, pediatric study subjects. Great. Moving on to challenge question number 2. The typical comment period for a draft PSG is A, 30 days, B, 60 days, C, 90 days, or D, 120 days. The correct answer is B, 60 days. As Dave mentioned at the beginning of his presentation, the learning objectives for this product-specific guidance sessions were to learn about the general principle of PSGs, along with a process of how PSGs are developed and revised with the recent examples. We hope that our presentations have provided some useful background information on how PSGs can help in generic drug development. To summarize, we showed you how PSGs and general guidance provide recommendations to efficiently develop generic drug products, how PSGs are developed based on public health priorities, requests from industry, current and anticipated patient industry needs, and on scientific research. With respect to our research efforts, it is our interest to advance the science of generic drugs and provide generic drug applicants and FDA the essential tools and information to help expedite the availability of generic drug products. We have ongoing efforts to update PSGs with the latest current thinking as new information becomes available. And lastly, there are additional resources such as control correspondence, pre-ANDA meetings, and plan new and revised PSGs for complex generic drug products to promote generic drug development. With that, this slide has some useful and relevant web page links for your reference. For more information about FDA's PSG publications and to search for most recent version of a PSG, see the product-specific guidances for generic drug development webpage. Thank you so much for your attention.
And thank you to Dave and MJ for those great presentations. We are about to go into our very first Q&A session, which means we're doing a little bit of dancing to get folks ready to roll in the background with their microphones all set and everything ready to go. Let me forward you, the participants, let me remind you that Q&A pod is down in the lower right-hand corner. So if you have a question specific for Dave, please preface your question with that @Dave symbol. If you have a question for MJ, feel free to preface it with MJ. And we are collecting those questions in the background. And I believe we may have our first one ready to go. But to find out, I am going to turn it over to Lisa. Do we have a question ready?
Hi, Jeff. Yes, we have many questions. So our first question is for Dave. If a product-specific guidance is issued for a specific product, would the FDA be able to answer questions in a controlled correspondence about bioequivalence?
And maybe we're having a little bit of trouble with Dave's microphone. Dave, are you up at the top of the screen, little green microphone next to it?
Yes, a controlled correspondence would be the appropriate mechanism to contact FDA about the recommendations.
Okay, great. So controlled correspondence it is. All right. Our next question is for Myungjin. Is the Fed study, in addition to the FASTED study, a new requirement, or is it only a recommendation? We'll just wait for the audio to come up.
And it looks like maybe we're having a little bit of trouble with MJ's audio. MJ, again, if you're got that green icon at the top of your bar menu screen there, next to it should be a little drop-down area where you
for
can...
For the modified release drug products, of course, you need to conduct Fed and FASTED BE studies. However, for the immediate release drug products, unless there are major safety concerns. Hello?
Hi, MJ. Do you think you could just repeat what you were saying? I think the first part might have gotten cut off a little bit. So if you don't mind repeating your answer, that would be wonderful. And the question is the Fed study, in addition to the FASTED study, a new requirement, or is it only a recommendation? Okay.
Can you hear me okay?
Now we can hear you. Mm-hmm.
Okay, for the modified release drug products, you should be conducting a fat and fasted BE studies. However, for the immediate release or also
Okay, maybe we want to go on to our next question for Dave. Are you ready, Dave?
So for the so I'm not sure whether you heard my response to the modified release products. For just once again, for the modified release products, you should be conducting a fat and fasted BE studies. For the immediate release products, depends on what the RLD label states. You may need to conduct fat BE study in addition to.
Yes.
Okay, and I'm going to interrupt. This is Jeff, and again, I apologize. We seem to be having some internet lag and congestion, but I am, now that we're both back, I'm going to go ahead and pass it over to Lisa. And Lisa, if you've got another question, you can direct to Dave or MJ.
Okay, sounds good. This next question is for Dave. For ANDA development of older non-complex products, where the product-specific guidance is not published, in that case, can they do a study similar to an approved ANDA product and do the clinical studies for BE in that way? Right?
And greetings again. Obviously, we're having some issues with both the
For
audio
products
micro
that do not currently have a product-specific guidance issued, your applicants should still use the most accurate, sensitive, and reproducible approach. I mean, they can certainly look at other product-specific guidances that are for similar products, but I would recommend that if they have questions to contact FDA about the approach that they're going to use.
Okay, thank you, Dave. I'll just ask you another question then. So this one's kind of simple. Could you please repeat the email address for requesting a new product-specific guidance to the FDA?
Yes, so the email address is generic drugs, all one word, at fda.hhs.gov.
And that is, I apologize to all the participants. Obviously, we're working out the technology. We had some pretty big lag on the audio for some of our speakers. but I'm looking at the clock, and in order for us to stay on schedule, we are going to have to cut it off. We will be working on improving the Q&A portion. That's our first time out of the gate, so hopefully after the next two presenters, things will go a bit more smoothly. But I do want to thank Dave and MJ for connecting to us from their homes so that we can they could answer those questions after their presentations. With that, we are going to move on to our next presentation. So I'm going to turn it over